And now we proceed with the awards. The region will be in this competition. We have 3 tracks. Maybe in the next one there'll be other tracks, but in this competition there'll be 3 tracks. One is computational biology of aging and longevity, the track which is directly related to Joseph's works. The second is fundamental biology of aging and longevity, and the third is social gerontology. In each track we have several contestants. Not many. We would hope there were more, but hopefully in the next prize there will be more. Also, you know, the situation doesn't encourage, but hopefully next time we'll have more contestants. We will start with short presentations up to 5 minutes without questions by the contestants in each of the tracks. In the end of each track, we will announce the winners, the first or the second prize. All the works are great, amazing. All the contestants will receive prizes. I will start with computational biology of aging and longevity, and the first contestant is Neta Nevo from Shai Shen-Or's lab. She will speak about identification of drivers for accelerating the development of B cells in old individuals, and the co-authors include Neta Nevo, Neta Milman, Ayelet Alpert, Tim Cooper, Doron Melamed, and Shai Shen-Or. So please, Neta, we can close this.
Hi, my name is Neta. I'm a pediatric hemato-oncology physician, and currently I'm doing my PhD under the guidance of Professor Shai Shen-Or. My project deals with one of the challenging clinical problems that increase morbidity and mortality among old individuals, low immunity. And we focused on the B cells that are responsible for mounting an immune response to vaccines, infections, and cancers, and whose frequency in the peripheral blood is low among old individuals. And to address it, we identified putative drivers for accelerating the development of B cells in old individuals. B cells derive from hematopoietic stem cells in the bone marrow that differentiate into progenitor B cells. And this longitudinal process is regulated by specific cellular niches that include many mesenchymal stromal cells. The bone marrow includes thousands of factors that theoretically may manipulate the development of B cells. The space is huge. And our goal was to identify those with the high probability to enhance the process of B lymphopoiesis among old individuals. And so for that, first we identified putative drivers in healthy donors. Then we focused on those that are relevant for old individuals. And finally, we validated the drivers. So first, we analyzed single-cell RNA-seq data of healthy human bone marrow samples. And we created a B cell trajectory that ordered the cells based on their developmental state. And our trajectory successfully captured the known B cell developmental stages, started from hematopoietic stem cells that differentiate into pro-B, pre-B, and immature B cells. And we identified 2,618 genes that create the trajectory and govern its development, and we named them B cell developmental genes. And then we aim to find activator for those genes, since those activators are putative drivers. And for that, we studied the cell-cell interactions in the bone marrow. And we identified 69 putative B cell drivers, which are ligands that are expressed by mesenchymal stromal cells and interact with receptors that are expressed by progenitor B cells. And next, we aimed to filter those that are relevant for old individuals. And for that, we studied the process of B lymphopoiesis upon aging in 3 different aspects. We studied the longitudinal development of B cells upon aging and the different regulation of B lymphopoiesis by mesenchymal stromal cells and by the crosstalk between the peripheral blood and the bone marrow. So to study the longitudinal development of B cells upon aging, we compared the trajectories of individuals in different ages. And we found B cell developmental receptors with different dynamic along the trajectory. For example, here you can see that the expression of 604 is similar within young adult. However, the expression of SPN, its dynamic precedes in yang, and therefore we prioritize its ligand, ICAM1. And we also studied the different regulation of B lymphopoiesis by the peripheral blood upon aging. So for that, we measured the protein level in the peripheral blood of 3 groups of patients: young individuals, old individuals, and old patients that by treating them with B-cell depleted therapy, rituximab, we induced B lymphopoiesis. And we identified putative drivers whose expression post-induction of B lymphopoiesis, so all the individuals that were treated with rituximab, was restored to useful, suggesting the role in B lymphopoiesis, for example, PDGFB. So overall, we identified 27 putative drivers that are involved in B-cell-related interactions. And then we decided to further test the role of 7 candidates that they display different biological function, and the role in B lymphopoiesis has not been established yet. So for that, we performed a single-cell gene expression stimulation assay. We stimulated the bone marrow progenitor cells with the different putative drivers, and we looked at the resulting downstream pathways and genes, and we found that 5 out of our putative drivers elevated B-cell-related pathways and genes. And finally, we focused on 1 putative drivers, a neurotrophic factor, whose role in B lymphopoiesis has not been established yet, and we validate it in a biological system that mimics the development of B cells from hematopoietic stem cells. And following addition of BDNF, we found accelerated development into progenitor B cells that express CD10 and CD19. So to sum up, we used systems biology approach and biological experiments to find the B-cell drivers for accelerating the development of B-cells in the bone marrow. And this may improve the function of other crucial age systems as well and lead to healthy aging. And with that, I would like to end and thank for my dedicated supervisor, Professor Shai Shenor, and the other lab members that support me in this project. And thank you very much.
Thank you, Netta. The next presenter in this track is Eric Feldman, also from Professor Shai Shenor's lab. And he will speak about towards healthier aging, adopting an immune state metric to predict disease and treatment outcomes. And the collaborators include Eric Feldman, Tim Cooper, Netta Millman, Yuval Klein, Aviv Barak, and Shai Shenor. Okay, that's fine. That's absolutely fine. The award does not depend on presenting or not presenting, only on the actual material, on the actual scientific value. And the posters are available here in the lobby. That's fine. And the third person in this track is Omri Segev, who is not here once again because of active military duty, and his supervisor, Professor Tali Kukerman-Yaffe. And his topic was development and first stage validation of a digital version of the digit symbol substitution test for use in assessing cognitive function in older people with diabetes. And the collaborators are Omri Segev and Tali Kukerman-Yaffe. So we will now announce the winner in this track. The prize money will go to the contestants' bank accounts directly. As I mentioned, all of them will get prizes. Only the difference if it's the first or the second prize. Both are very honorable. So the winner is Eric Feldman. Welcome. That's the first prize. And the second prize goes to Neta Nevo. Thank you very much. We go to the second track, which is Fundamental Biology of Aging and Longevity. And the first contestant is Noa Rahmian from Valery Krzyżanowski's lab from Weizmann Institute. And her topic is the role of microglial senescence in aging in Alzheimer's disease. And the cooperators are Rahmian, Medina, Cherqui, Akiva, Deutsch, Dilby, Kreuze, Salame, Ramos, Cajalon, Krzyżanowski, Michael Schwarz, all from Weizmann Institute. Welcome.
Thank you. Thank you for the organizers and for Larissa for this prize. And my name is Noa Rahmian, as you said. I'm going to present to you my PhD project. It was published in Nature Neuroscience about the connection between senescent microglia accumulation, aging, and Alzheimer's disease. First, a bit of an introduction. Alzheimer's disease is a chronic neurodegenerative condition marked by brain inflammation and cognitive decline. Microglia are the innate immune cells of the brain, the major immune cell of the brain. They are having a significant role in Alzheimer's disease. Aging is a major risk factor for Alzheimer's disease, and it's categorized by the accumulation of senescent cells in different tissues, contributing to the chronic inflammation in aging. Senescent microglia are the potential connection between aging, inflammation, and Alzheimer's disease. Yet, it's still not fully understood how. In my project, we showed that there is a conserved signature of senescent microglia across aging, Alzheimer's disease, and dementia. This signature, this conserved senescent microglia are accumulating in aging and in Alzheimer's disease mice, 5X mice. They also upregulate TREM2 expression, and TREM2 expression was correlated with senescent markers. When knocking out TREM2, there was a significant reduction in the senescent microglia accumulation. And when using a drug, ABT737, which causes senolytic effect of senescent cells, meaning it kills senescent cells all over the body, including the senescent microglia, there was an improvement in cognitive measurement using the novel object recognition task. There was less accumulation of senescent microglia and less neuroinflammation. So to sum up this project, aged mice and Alzheimer's mice accumulating microglia with a conserved signature. These senescent microglia are correlating the expression of senescent markers with TREM2 and ApoE that I didn't show you, but it's in the paper. When knocking out TREM2, we see less accumulation of senescent microglia, and using senolytic treatment ameliorated Alzheimer's disease pathology. Thank you.
Thank you very much, Noa. The next presenter in this category is Yulia Mayevska. Is she here? She's here. The supervisor is also Valery Krzyżanowski, and her topic is P16 dependent increase in PD-L1 stability regulates immunosurveillance of senescent cells and promotes chronic inflammation and aging. And the co-authors are Yulia Mayevska, Amit Agarwal, Avimayo, Urialon, Ido Amit, and Valery Krzyżanowski. Welcome. You should see this.
Okay, thank you very much to organizers for this kind introduction and invitation. Great. So senescence is a state of irreversible growth arrest that is induced by damage, and simply these cells overexpress inhibitors of cell cycle progression, and they don't die. They are metabolically active. They secrete a wide array of cytokines, calling immune system to come and kill them. However, this process is really disabled in aging, and they accumulate in aged individuals, leading to different age-related diseases. And we already know that elimination of these cells can improve both health span and increase lifespan. So this attracted lots of attention into development of senolytic drugs. As Noah said, that kill senescent cells. However, they are not specific yet, and they have side effects in human. And this was my question. So maybe it's worth to understand why do they accumulate? What's the molecular mechanism of accumulation that could lead to better strategies? So if you think about senescent cells accumulation, we need to think about turnover and dynamics in the body. Well, it depends on damage that leads to production of the cells and immune-mediated removal. However, in age, it's disabled because there's higher production rate that leads to higher production of the senescent cells, and immune system is also dysfunctional and doesn't remove them well. But what was not known was this interaction between senescent cells and the immune system itself. And I asked myself whether there is some intrinsic mechanism that can regulate the removal, whether we can target, and what would be the output. So to make long story short, to address all these questions, I wanted to understand cellular senescence at single cell level in lung as a model organ in both aging and chronic lung disease. And what I noticed is that myeloid cells were enriched, especially alveolar macrophages were enriched in senescent phenotype. And they were expressing high level P16 as inhibitor of cell cycle progression, but also the same cell type was very highly expressing immune checkpoint protein PD-L1. And this attracted our attention because PD-L1 is critical central protein controlling immune responses, overexpressed on cancer cells, leading to their evasion from immune clearance. So what we've seen, we studied it in detail, and we've seen that these cells that highly express P16, they were also highly expressing PD-L1, regardless of age, but it was more pronounced in aging in both population level and single cell level, and was independent of the cell type. Simply this phenomenon was in any cell that entered senescence. So the next step, I wanted to understand whether there is some molecular mechanism behind this process, and well, indeed, when I overexpressed P16, it led to accumulation of PD-L1, but the depletion of P16 caused accelerated rate of degradation. And this suggested that in this whole process of PD-L1 accumulation, there is aberrant degradation rate, and simply proteins that are involved in targeting PD-L1 for ubiquitination and subsequent degradation are aberrantly expressed, leading to less ubiquitination and accumulation of this protein. Well, so when we have PD-L1 on the surface, it can lead to suppression of immune system responses, but there are ways to go about it. And we can inhibit this axis using antibodies either against PD-L1 or PD-1. And this is indeed what I used, what I checked. However, for PD-L1 part, I use activating antibodies that not only block the axis, but also activated Fc gamma receptor positive cells towards killing PD-L1 positive cells. And simply what we've seen is this very interesting that only blocking PD-L part led to depletion of senescent cells in short-term model of lung inflammation. And it corresponded well with enhanced immune activity, cytotoxicity of CD activation of CD8 T cells. And the same phenomenon we observed in physiological models of aging and also chronic lung disease. So we could deplete senescent cells. It was related with T cell response, activation of T cell arm, and reduce chronic inflammation, systemic inflammation. So it all leads to simple conclusion that the damage that leads to induction of cellular senescence also leads to via P16-dependent pathways to decrease of ubiquitination of checkpoint, PDL1, and its induced stability. And when it accumulates and is upregulated on the cellular membrane, it leads to it shuts down immune response, leading to evasion of the cells and accumulation in the body, but we can target it, and elevation of inflammation and different senescence-mediated age-related diseases, but we can target it with antibodies against PDL1, which can enhance immune system to come and kill, remove senescent cells in vivo, and simply ameliorate inflammaging. And this potentially opens, whole strategy of designing anti-aging drugs that can be based on immunotherapy. Thank you.
Thank you, Julia. And the next contestant, the final contestant in the fundamental track is Ekaterina Rudnitsky from Professor Vadim Freifeld's lab in Ben-Gurion University in Beersheba. And she will present on the impact of MSC-derived extracellular vesicles on the crosstalk between senescent and stem cells. And the collaborators include Ekaterina Rudnitsky, Gadi Turjiman, Vera Garbunova, Marina Wolfson, Hachik Muradyan, and Vadim Freifeld. Welcome.
Okay, first of all, I want to thank organizers for awarding our works and for this opportunity to present our results. At this conference, we heard a lot about cellular senescence and presumably about and predominantly about a negative side of senescent cells. They, however, play a dual role in biological systems. Indeed, in recent years, cellular senescence was suggested to be an important or even obligatory phenomenon in cell reprogramming. But what's about natural stem cells? Indeed, in our lab, we found that the number of stem cells in fibroblast cultures depends on the stage of cellular senescence. All this points towards a possible crosstalk between senescent and stem cells. Among different ways of cell-cell communication, the important role belongs to extracellular vesicles. Our recent analysis clearly showed that mesenchymal stem cell-derived extracellular vesicles can mitigate the cellular senescence phenotype. And here, in this work, we report for the first time that impact of mesenchymal stem cell-derived extracellular vesicles on the pool of stem cells in senescent cultures. For this purpose, we collected extracellular vesicles from naive mesenchymal stem cells as well as anti-inflammatory polarized mesenchymal stem cells. And we supplemented them to incubation medium of human fibroblast cultures at various stages of cellular senescence. On the wall, we found that mesenchymal stem cell-derived extracellular vesicles attenuate cellular senescence. That is, reduce the percentage of SA beta-gal positive cells as well as multinucleated cells in culture, increased cell growth, and accelerated in vitro wound healing. As to stem cells, only extracellular vesicles from naive mesenchymal stem cells displayed a clear effect. As you can see in the graphs, these cells increased the number of stem cells in presenescent as well as senescent cell cultures. This elevation coincides well with the exosome-induced increase in still, with exosome-induced increase in senescent cells in pre-senescent fibroblast cultures, which we can see here. But the problem is here, like we see the decrease of senescent cell in the culture with the same time increase of these stem cells. At the moment, we don't know how to explain this discordancy in exosome effects. However, maybe it may be the matter of nonlinear relationship between cellular senescence and stemness. Whatever, this observation led us to the suggestion that mesenchymal stem cell-derived extracellular vesicles affect not only the pools of senescent and stem cells, but presumably also crosstalk between them. Thank you very much for your attention.
Thank you very much, Ekaterina. And these were all the contestants in the Fundamental Biology of Aging and Longevity track. And the winner of the first prize is Yulia Maevskaya. Welcome. Thank you. And the winners of the two winners of the second prize is Noah Rahmian. Thank you. And Ekaterina Rudnitsky. All right, so we go to the final category, which is social gerontology. The first contestant is Sanjuti Roy, supervised by Liat Ayalon from here, from Bar-Ilan University. And the topic is ageism as a barrier to health longevity. Is Sanjuti here? Not here? Here? Ah, do you want to present? No. That's fine. Once again, it's all about science, not presentations. And the second contestant is Peter Soon, also from the group of Liat Ayalon in Bar-Ilan University. And his topic is advancing longevity via activity engagement, multi-channel sequence analysis. Do you want to present? No? Once again, this makes us life easier and closer to the finish. So the winner of the first prize is Peter Soon. But you're also co-author with Sanjuti, so that's fine. And the second prize is accordingly goes to Sanjuti Roy. Thank you very much to all the contestants of this first round of Longevity Aspiration Prize. Thanks to Larissa, thanks to the founders of the Joseph Gitters Foundation in his memory. That will be hopefully a combination of several foundations, several support sources. The entire support, we don't divide the entire support for the Longevity Nation, conference goes through vertical association in cooperation with Bar-Ilan University, Department of Science, Technology, Society. The main sources of funding included also the Segol Network, American Federation for Aging Research, Shlomo Tyron Foundation, Deep Knowledge Group, Quadrascope, several other funders and supporters. We're grateful for all of them that even in this difficult time, they support this critical area of human development and of social and human endeavor. So we still have a formality. You know, we usually conferences adopt some kind of resolution. Generally, the entire purpose of this conference was to promote longevity field, health longevity field in Israel. Research, development, and education to raise this topic to the public awareness and academic awareness, to bring the attention to the attention of decision makers, supporters, funders, you know, to kind of increase the interconnectivity in the field, the matchmaking. So all this allows us to adopt some sort of a resolution, once again with the purpose to promote the field further. And the resolution can be the same as the last year Longevity Nation resolution. Basically, it repeats the main paragraphs, the main points of the section called Enhancing Research, Development, and Education for Health and Longevity and Prevention of Age-Related Diseases, which was actually adopted in 2019 by Knesset as part of the Israel National Master Plan on Aging report. It was published. Not many people remember it, but it's there. It's on paper. So, but we remember it. We also remember it in our conference. That's the name of this conference, Enhanced Research, Development, and Education for Health and Longevity. The main points include increasing funding, R&D budget for health and longevity research, obviously, the main necessary condition for this field to go forward. Establishing, improving a public health system for early detection, meaning basically developing metrics that we also discussed here a lot, metrics for health and longevity, as predictors of aging trajectory, predictors of aging health, as well as to evaluate the effectiveness of anti-aging lifespan and health span improving treatments, and also education, you know, teaching about the field of biology of aging in relation to other areas, because obviously the field is very multidisciplinary. Basically, every area of human endeavors can be somehow related to aging. Aging is life, but at a later age. So everything is related to aging. So basically, these are the 3 points of this program adopted by Knesset that we want to remind, and these are also the 3 points of our resolution. They were adopted in the last year. This year also said to all the speakers there were no objections, so we can say that this program is adopted as well. Unless anybody has objections, please talk with me. Once again, we must say thank you very much to all the participants, all the supporters, all the amazing lectures, the real cutting edge of longevity research, not just in Israel, but around the world. And we hope that Israel will take its rightful leading place among the leading nations, so in the longevity field, research, development, education, as well as enhancing cooperation with the whole world. And hopefully the situation will improve also security-wise, and we will be able to do more of this in the next year. Thank you very much again. So basically, unless somebody wants to raise some burning issue, no. Then we can have our lunch. Thank you.